In an institutional psychiatric setting, a specific profile emerges in the case notes of long-term patients. The charts apply a familiar set of labels to describe these individuals: non-compliant, lacks insight, withdrawn, or exhibiting poor engagement. Clinicians typically treat these behaviors as direct symptoms of the patient's underlying pathology, identifying the individual as the source of the resistance. Modern neuroscience identifies these behaviors as state-dependent biological adaptations rather than fixed character traits or symptoms of illness. These resistant behaviors reflect the nervous system reacting to the coercive architecture of the clinical environment itself. In operant conditioning, a basic biological rule applies. Behaviors are shaped by their consequences. Reinforcement increases the frequency of an action, while punishment decreases it. Institutional settings frequently violate this rule. A patient's honest disclosure of distress or suicidality is routinely met with punitive measures, ranging from sudden seclusion to a loss of ward privileges. This unpredictability destabilizes the nervous system. The patient cannot reliably predict whether a cry for help will result in medical care or institutional punishment. When an institution punishes honesty, withdrawal and strategic silence become the biologically accurate adaptations to inconsistent contingencies. The human brain operates as a predictive inference machine. It updates internal models of cause and effect to navigate the world, conserving metabolic energy. When clinical contingencies become unstable, the outcomes patients experience become weakly linked to their actual behavior. Computational neuroscience defines this as the credit assignment problem. The signal-to-noise ratio in the environment degrades. The brain ceases to map its own actions to reliable outcomes because the environment has randomized the results of those actions. Once the brain can no longer assign credit to its own behavior, the nervous system abandons pro-social, help-seeking actions. This explains the profound passivity and withdrawal often observed in long-term institutionalized patients. While early theories called this learned helplessness, contemporary neuroscience has inverted that model. Passivity exists as the brain's biological default when exposed to prolonged aversive conditions. Control is the specific state the brain must learn to acquire. The ventromedial prefrontal cortex, or VMPFC, functions as a control detection circuit. When it senses agency in the environment, it inhibits the body's stress responses. When experiences indicate that outcomes are uncontrollable, this control detection system fails to engage. The calming signals stop. Clinical withdrawal reflects a failure of the VMPFC to engage when an environment provides no evidence that a patient's actions influence their outcome. Under chronic institutional threat, routine procedures like footsteps, formal social evaluations, or mandatory check-ins become active threat cues. Executive regulation and threat response are physically linked by the uncinate fasciculus, a white matter pathway connecting the prefrontal cortex to the amygdala. When triggered by an temporarily weaken the dorsolateral prefrontal cortex. The dorsolateral prefrontal cortex supports working memory, cognitive flexibility, and deliberate emotional regulation. The behaviors recorded as concrete thinking or a lack of insight are biologically determined by the stress-induced shutdown of the prefrontal cortex during clinical evaluation. When an overt danger or a coercive event has passed, the patient's nervous system often fails to generate a safety signal. The hippocampus is responsible for context discrimination. It encodes when and where events occur, separating a dangerous then from a safe now. The hippocampus is highly sensitive to stress hormones. Prolonged exposure to elevated glucocorticoids causes complex dendritic branches to physically retract. These complex networks are stripped down into rigid, simplified pathways. This physical dendritic remodeling biases the brain away from flexible memory strategies toward habitual defensive responses. The remodeled hippocampus lacks the physical capacity to distinguish a current benign clinical check-in from the intense previous experiences of institutional trauma. While these anatomical changes are severe, they intersect with the brain's inherent capacity for neuroplasticity. Many stress-related neural injuries, including hippocampal remodeling, show evidence of partial reversibility. Biology requires specific conditions for this reversal: a state of sustained safety, reduced environmental stress, and reliable behavioral contingencies. Talk therapy remains biologically ineffective if the underlying institutional environment remains coercive and unpredictable. Expecting therapeutic insight is biologically inconsistent with an environment that repeatedly triggers survival circuits. To achieve different outcomes, the burden of change must shift from the patient's brain to the institution's policy and design. Policies must ensure that a patient's honesty and disclosures of distress are met with consistent, non-punitive support. Pro-social behaviors must be reliably reinforced to repair the brain's predictive processing models. Consistent, non-punitive interactions with clinical staff provide the brain with the long-term safety signals necessary for biological and emotional regulation. To stop producing treatment-resistant patients, psychiatry must abandon administrative coercion and rebuild the institutional architecture to foster biological safety.